We will fly until Mendoza, Argentina, and say good morning to Sofia Masuelli, who will present the work entitled When Left Does Not Seem Right, Epigenetic and Bioelectric Differences Between Left and Right Side Breast Cancer. Sofia, are you there?
Yes, hello.
Good morning.
Can you hear me okay? Hello?
Yes, we can hear you.
Okay, thank you. Well, thank you very much for the introduction, and I'm very glad to be here. Well, first, I will start saying that according to the 2020 Global Can data, breast cancer has a worldwide incidence of more than 2 million women per year. Also, during tumorigenesis, normal cells acquire genetic and epigenetic modifications to turn into cancer cells. Among the epigenetic modifications, DNA methylation is the most studied process in cancer cells. On previous studies of our group, an interesting finding revealed that left and right tumors have differential methylated profiles. And well, for this talk, I need to introduce you to this concept is bioelectricity. Well, our cells are electrically active, and they have a bioelectric circuit that generates membrane potential and internal bioelectric states that affect communication and also cell functions like proliferation, migration, differentiation, and gene expression. So, well, the previous result on finding that I told you about differential methylated on left and right breast tumors generate a lot of questions for us, and so we propose a hypothesis to understand it. Since it is known that left and right breast glands are non-identical, we propose that tumors growing in the left gland establish a different communication with the environment as compared to the right-sided ones. And also, bioelectric and epigenetic changes have a crucial role in this different communication. Well, to study this hypothesis, we use three approaches. First, we use an in silico model in which we analyze data from public database like TCGA. We also use an in vitro model in which we incubate breast cancer cell lines during 5 days with left and right extracts obtained from mammary reductions of healthy donors. And we also use an in vivo model in which we inoculated breast cancer cell lines into left and right glands of mice, immune-suppressed mice, to generate bilateral xenografts. These are our results, and well, we started collecting data from breast cancer TCGA, and we selected the 169 genes with left-right differences above 3%. By enrichment analysis, we observed that most of these genes were associated to embryogenic and neural type features. We also want to see if this was reproducible in vivo, so we did this bilateral xenograph that I mentioned before, and we analyzed the methylation differences in 3 passages, and we chose the one with the highest difference. These tumors were analyzed, we analyzed their methylome by RRBS, and we select the top 1,200 genes with left and right different methylation. And by enrichment analysis, we observed again that these genes were associated to embryogenic and neural type features. In these both features, ion channels are crucial regulators. So we search if we have ion channels in our differentially methylated list. In silico list, and in our in vivo list. Also, dichotomizing their function in hyperpolarizing channels and depolarizing channels, we observe an increased percentage of methylation in hyperpolarizing channels in left tumors. This suggests maybe a more depolarized state on left tumors. Next, we want to test these bioelectric differences. So we use the in vitro model that I told you before with the left, the breast cancer cell lines treated with the left and right extracts during 5 days. And we measure calcium concentration with a specific probe by confocal microscopy. And we observe an increased calcium concentration in left treated cells compared to the right ones. This is in line with a more depolarized state on left treated cells. Next, with the same methodology, we analyze the membrane potential with a specific probe by flow cytometry. And we observe an increased membrane potential in left-treated cells compared to the right ones. We use 3 different extracts from different donors, and we observe the same result, but when we mix the 3 donors and in pools, left and right, we observe that these differences increase. So we decided to use pools to avoid this specific donor bias. And we also want to normalize our results. So we use a depolarizing agent to totally depolarize cells, and we observed that left-treated cells did not differ significantly to the totally depolarizing cells. So these results confirm that left-treated cells and left tumors have a more depolarized state. Well, switching again to the in silico model, we want to see if left and right tumors have differences of epigenetic regulators. So we analyze data of gene expression of the DNMTs and TETs, these enzymes, epigenetic regulators, and we observe an increased expression of DNMTs in left tumors, and we observe no differences in TET enzymes between left and right tumors. Also, we wonder if these epigenetic and bioelectric differences that we have observed have any relation with a functional feature like proliferation. So we analyze in silico data of gene expression of T67, and we observe increased expression of T67 on left tumors, and in concordance, we observe a higher mitotic count in left tumors from our database of local patients. So with all these results, we can conclude that left-treated cells or left-sided tumors present increased methylation and a more hyperpolarizing ion channel genes, and consistently show a more depolarized membrane potential and increased proliferation rates as compared to the right ones. Also, we think that epigenetic has a crucial role in these lateral differences, and our work could serve as proof of principle for other bilateral cancers. And well, all these results left us with new questions that we want to answer soon. But well, I am going to tell you some of them. Maybe first, does it occur, these changes, bioelectric and epigenetic changes occur in a specific order? And if so, what occurs first? If bioelectric changes are generating the epigenetic modification, or is in the other way? And also, if is it bidirectional? And well, we also want to know what components have these left and right microenvironments that are generating these bioelectric and these epigenetic changes. Okay, thank you, and thank you for the invitation. Hello. Are you there?
Yes, yes, we are here.
Okay.
Thank you so much. The paper is open for discussion. Any question? Well, I was very curious about the extracts you mentioned from the patients. Could you tell us a
Yes.
little bit more, because well, it's new for me.
Yes. Well, we obtain tissue, mammary tissue, normal mammary tissue from donors that did surgeries or mammary reductions. And well, we mix this tissue with a little of medium and let them to liberate all these. Sorry, I don't remember the word. So we'll liberate all the factors that they have, and during 24 hours, and then we use a proportion of 50% of these extracts with 50% of the normal medium, because we observed that if we use only the extracts, cells didn't survive, so we decided to use 50%.
Okay, so it's conditionated media. No?
Yes, it's conditioned.
So,
yes.
And at the clinical level, maybe I miss it, but are differences between the frequency between left and right?
Yes, the differences between left and right are subtle, but are consistent. And well, there is a higher incidence of left side left tumors, but it's very subtle. It's 51%, but it's consistent. And so, well, it's the same with our results. These results are not big differences, but they are consistent with every patient, every donors that we use. We use we did on mice also, and we saw the same.
Well, thank you very much, Sofia, for your talk. And
okay, thank you.
And well, thank you for participating also from so far away. And I hope you
so
enjoyed the meeting. Okay, we will.
Okay. Okay, thank you very much.
Okay, bye. Bye.
Bye bye.